Hello students, welcome back to our biology class. Myself Dr. Raghavendra here. Well, in the earlier classes we studied lots and lots of important chapters. Yes, we discussed about the animal kingdom. Yes, we would have studied about even the structural organization in animals now. So, the next important chapter I am going to discuss with you is a very essential chapter. Yes, it can be a common in first year biology as well as in the second year chemistry also. You will be learning about that. Title of the chapter today what I am going to discuss with you. Yes, it is the biomolecules. So let us go ahead with the chapter biomolecules. Fine. So the chapter biomolecules I am going to discuss with you people. Biomolecules. When the term we tell biomolecules, what come to your mind? Biomolecules. Bio refers to be the living molecules, means yes, they are the chemicals in fact. So the chemicals produced in the living cell as a result of metabolism, we call it as a biomolecules. Yes, under this chapter we will be discussing more about the what you can say. Number one, the fact as such, analysis of biomolecules. We are going to discuss biomolecules. Secondly, we will study about the primary and secondary metabolites. We will be discussing about this, right? And thirdly, we will be discussing about the biomolecules as such. Third, molecules. So types. Under this, we will study about structural units of biomolecules, and then physiological importance of biomolecules. Physiological importance of biomolecules means we will be discussing about how the proteins are made up of, how the carbohydrates, how the lipids, how the nucleic acids are made up of. We are going to study in detail about all this, the structural aspects, and then their physiological importance also we will be discussing. Then we will be discussing about, most importantly, what is metabolism. What is metabolism? And then what is the living state? Living state means the metabolism always occurs in our body. It is mainly cutting up of reactions. We can say that the complex food we eat. Further, the food is a composition of mainly, it may be the proteins, carbohydrates, lipids, and then number of other things will be there. So proteins will be broken up into amino acids, carbohydrates will be broken into the simple sugars, lipids will be broken into the fatty acids. Like this breaking mechanism we call it the catabolic reaction, or the glucose combined together it becomes the glycogen. So it is anabolic reaction, means from simple to complex molecules are made and they are stored in our body. Okay. So the total of these catabolic and anabolic reactions together we call it the metabolism. So metabolism happens only in the living body, not in the dead body. So in our blood, sugar level is measured in terms of mgs, hormone level measured in terms of the nanograms, we can say that. Like that, every biochemical component has its own optimum range. So all are not at all equal, they are of different state. So different levels, different range of biomolecules are measured or maintained in the body. So unequilibrium state is a kind of the synonym to the living. So all the living activity happens to maintain the different optimum range of different molecules. All the molecules cannot reach the same point. If equilibrium reached, that is not a living body, it may become the dead body. So in the physics also it said that at equilibrium state no function is carried out or done. So on equilibrium state only there is a here and there movement or function is going to happen. Well, So we are going to study about what is metabolism, what is the living state also. Then we are going to discuss more about the enzymes. Enzymes, you know that they are the biocatalyst in fact. So enzymes known to mainly induce or increase the rate of reaction. Rate of reaction, how actually enzymes influences. Then we are going to discuss about the factors influencing enzyme reaction factors, and then we are going to discuss about classification of enzymes. Classification of enzymes we will be discussing. Other than that, yes, we are also studying under the chapter biomolecules. What do you mean by cofactors? Cofactors, they can be prosthetic groups, and then there is what we can say that yes, they can be the coenzymes, they can be the metal ions. So these are all the important things. Yes, we are going to study one by one under the chapter biomolecules. I hope you got an outline or clarity about this chapter. Yes, under the chapter biomolecules we are going to study analysis of biomolecules, primary and secondary, what are the metabolites we are going to discuss, biomolecules types. As such, we are going to discuss four important types. They are proteins, carbohydrates, they are the lipids, as well as nucleic acids. So how these biomolecules are made up of? What is the structural unit in them? Proteins have the amino acid as the structural units. Okay. Carbohydrates have the simple sugars as structural units. Lipids have the fatty acids as structural units. And then nucleic acids have nucleotides as their structural units. We are going to study all of that. Now, when we talk about mainly the biomolecules, let me give you the definition of biomolecules. So, biomolecules as such, biomolecules are the chemical components of the living cell and are produced as a result of metabolism. So cell metabolism is very, very important. And biomolecules, yes. So when you talk about biomolecules, whether the variety of living creatures you consider, whether you take the plants or the animals or the microorganisms, so all of them have the common type of what we can say biomolecules. So all of them have the biomolecules. Yes, you can find the biomolecules such as the proteins, carbohydrates, then lipids, as well as nucleic acids. Yes, these are the basic type of biomolecules in all these plants, the animals and microorganisms. That includes the bacteria, fungi also. Okay, so we find the basic type of biomolecules in all these. And basically biomolecules are made up of what? Yes, biomolecules are made up of, we can say that they are made up of carbon, then hydrogen, oxygen, nitrogen, and even phosphates, the phosphate group also we can say. So like that, biomolecules, yes, they are definitely made up of these variety of simple compounds. Generally, the living organisms, yes, they have arrived from, we can say that earth itself. So earth as well as the living cell we consider, lots of common chemicals we can find in between them. Consider, yes, by weight the percentage of different kind of biomolecules as such. So when we consider mainly the different type of biomolecules, yes, we can find lots of common factor. So thus we can say that the earth's crust and living cells have lots of common, we can say that common elements or the chemicals. So this is what we called as the similarity between the non-living as well as living. Definitely non-living also has certain compounds, even living cells also have number of certain compounds. So thus we can find mainly variety of these compounds in the living as well as in the non-living. Say for example water. Yes, it is there in the earth also. Yes, we do have also in every cell we have the water. So carbon, hydrogen, oxygen, nitrogen, yes, all these we can find in the non-living also as well as in the living also. And one more important thing we can say that biomolecules, they are basically, we can say that they can be inorganic, they can be organic. So inorganic compounds without carbon in them, so they are without carbon. Organic compounds with carbon. Okay, yes. So inorganic compounds, it may be the sodium, potassium, calcium, magnesium, water, all these are inorganic. When we say organic compounds, whether you consider the proteins, carbohydrates, lipids or nucleic acids, everything on this earth which seems to be the living, they will have definitely carbon in them. So we call them as organic compounds. So biomolecules can be inorganic as well as organic. So we need to understand what are those important chemical components that can be very much seen in the earth crust as well as in the human body. Let us discuss that. So considering those things, I hope you got a clarity. Biomolecules are those compounds or the chemicals synthesized by the living cell. Okay, and they are basically made up of carbon, hydrogen, oxygen and nitrogen. So we will understand how there is a similarity between the earth crust and a living human cell also. So yes, let us understand. When we say that mainly, so the chemicals, yes, and then percent weight, yes, by weight we can measurable, they are measurable mainly in the earth's crust as well as the human body, yes, by percentage wise. So when we say nitrogen in the earth crust, very little, and in the human body, yes, nitrogen is there as such somewhere around 3.5%, we can say. And then hydrogen is there in the earth crust, yes, we can say that somewhere it is around 0.14%, and here it is somewhere around 0.5. Then oxygen, yes, in the earth crust we can find 46.66%. In the human body it is 65.00. Okay, then magnesium, it is 2.5%. Yes, we can say that somewhere it is here 0.1%. Okay, then we can say that carbon, it is 0.03%, whereas 18.5% is the carbon in the human body. And then calcium, okay, calcium, yes, we can say that it is somewhere around 3.5. In the human body it is 1.5 calcium. Then again the next is the sulfur. Yes, in the crust we can find the sulfur 0.03%. In the human body it is 0.3%. And sodium, yes, sodium we can find as such 0.28%, whereas in the earth crust we can find it as 2.8%, whereas in the human body, yes, we can find 0.2%. Then silicon can be seen as 27.7% in the earth crust, but in the human body we will find it very, very negligible. So these many chemical compounds, yes, that can be seen in the earth crust as well as in the human body. And this is what we say how the living organisms have been arrived from the non-living only, and that's why they indicate the existence of a similar kind of chemicals between what we can say mainly earth crust as well as the human body. I hope you are understanding these important points. So that's what we can say that mainly here. So this is all about mainly the kind of chemicals that can be seen in the earth crust as well as in the human body. Nitrogen is very little in the earth crust, but nitrogen is very, very important to make whether it's a DNA or any other chemical substances. Yes, nitrogen is required. So 3.5% in the human body we can find it. We will find the hydrogen 0.14% in the earth crust, 0.5% in the human body. Oxygen 46.66. Our body requires more amount of oxygen to carry lots of metabolic activity. So 65%, yes, we do have the oxygen. Magnesium 2.5%, it makes 0.1% in the human body. Carbon 0.03 and 18.5% in the human body cells, yes, we can say that. Then calcium, yes, 3.5, 1.5% in the human body cells. Sulfur 0.03 and 0.3% in the human body cells. Then sodium 2.8 and it is 0.2 in the human body cells compared with the earth crust. So silicon is plentifully available in the earth crust 27.7%, whereas negligible amount will be there in the human body cells. This is what we can say the similarity between the availability of the certain type of chemicals between the human body cells as well as the earth crust. Well, we need to understand, as I told you, yes, definitely our body cells not always having the organic compounds. There are certain inorganic compounds also, yes, we do have. Let us study that also. So what are those different types of inorganic substances? So let us understand that also. So this is what we talk about mainly, the similarity or comparison between the earth crust as well as the human body cells. When we talk about inorganic mainly compounds in the human body cells, inorganic compounds. So these are the compounds without any carbon in them. So they can be as such sodium, potassium, then calcium, magnesium, okay, then availability of the water and other compounds. So how they are mainly represented? Sodium we call it as Na plus, potassium Ka plus, calcium Ca plus, magnesium Mg plus plus, water denoted as H2O. Other compounds means it may be calcium carbonate, it may be MgCl2, it may be NaCl. Okay, then there may be what we can say that the phosphate group, then it may be the sulfur. So this is what we talk about, different kind of inorganic substances that also do exist in the living tissues or living bodies. So they are denoted by different kind of formulae. This is what we talk all about mainly, the types of biomolecules. So let us understand how we are able to measure all these and how these biomolecules are measurable also. Yes, there are the methods or the technique what we are going to study, that is called as analysis of biomolecules. Let us understand the analysis of biomolecules. Fine. So when we talk about, yes, inorganic compound means the compounds without any, yes, carbon in them. So what we have so far discussed, those are all the compounds without any kind of what we can say the carbon, such as sodium. Yes, you can't find the carbon in that. Then maybe what we can say the potassium, sodium, potassium, then calcium, okay, then magnesium, fine, then water as well as other compounds. It may be the calcium carbonate, it may be sodium chloride, it may be magnesium chloride, it may be the sulfates as well as phosphates. You won't find the carbon in them, that's why they are called as inorganic compounds. But still the inorganic compounds are very essential for the living cell, okay, to carry out number of metabolic activities. Now let us understand mainly what is that, the analysis of biomolecules. So we are going to understand mainly analysis of biomolecules. How we are going to isolate these biomolecules? So let me tell you, it is very simple. Yes, consider you are taking the tissue. So the tissue, it may be the plant or it may be the animal tissue or it may be the microbes. Okay, the clump of microorganisms. Then what you are going to do with this tissue? So add, what we are going to add, trichlor acetic acid, what we call it as the TCA. So it is also called as TCA. It is denoted as Cl3 CCOOH. Okay, trichloroacetic acid. Okay, and then plus the tissue. Okay, what you are going to apply now? You are going to do the grinding using mortar and pestle. What is this mortar generally? Yes, it is a ceramic device and here is the pestle. Yes, with this we are crushing all the tissue, mortar and pestle, grinding. Further, yes, we are filtering now. Filtering by using cheesecloth. After filtering, yes, we get mainly two important components. So it is called as acid soluble pool. Another acid insoluble pool. Okay, these are the two important things, yes, we are going to get after filtration. So acid soluble pool mainly, it is a cytoplasmic content. cytoplasmic components. Okay, yes, here it is mainly cell membrane components. Okay, so thus acid soluble and acid insoluble pool we get. So what we are going to do further? Yes, we are collecting that all in a test tube, and yes, what we find here. So this is the component biomolecules or acid soluble pool. So what it contains? It contains mainly again proteins, carbohydrates, lipids as well as nucleic acids. Yes. So generally lipids are acid insoluble pool they form. Okay, yes. So what actually lipids construct? Lipids forms mainly membranes of organelle and plasma membrane also. Plasma membrane. Fine. So this is what we talk about mainly in a test tube. If you consider the biomolecules, yes, they all are the acid soluble pool that has mainly the cytoplasm component. In the cytoplasm we find the proteins, carbohydrates, lipids and nucleic acid, but majority of the high molecular weight lipids will become insoluble in the acid pool, and yes, they are mainly from the cell membrane as well as membranes of the organelle. So thus ultimately by mainly getting those biomolecules, yes, by adding number of chemical reagents, number of biochemical tests, we can analyze what is the quantity of these biomolecules. Microgram per ml or microgram or milligram per deciliter or 100 ml, yes, we can analyze. This is what we can say mainly the method of analysis. So there is again one more method of analysis. What is that? The one more method of analysis, it is mainly by burning the mainly organic matter, turning up into the ashes, and those ashes are collected and made into small pellet through which atomic absorption rays are passed. As a result, we can get the different peak of the chemicals, and with that we can analyze even chemical structure of that particular content or compound, and what compound it is, how many components are there in that compound also in detail we can analyze. So that is called as the atomic absorption spectroscopy. Yes, that we will discuss. Let me tell you once again, if you have the material, it may be the leaves of the plant, or it may be the liver of the animal, or it may be the clump of microorganism, you are taking it and crushing it by using what? Trichloroacetic acid. Why they have used TCA? Because it is mainly the acid that disintegrates the tissues, separates the cells, then crushing such separated cells make very easily outflow of the biomolecules. So the outflow of the biomolecules, further what we can say that it is filtered. What actually it happens means mainly filtering by using the cheesecloth. So what we generate, it is called as a slurry. The gel-like substance or slurry developed filtering that, then we get the acid soluble pool, acid insoluble pool. In the acid soluble pool, we will be having all the content of the cytoplasm. Acid insoluble pool, we will have the content of the cell membrane as well as the membranes of the organelle. And then in a test tube, when we can mainly collect all those, further we apply either centrifugation, or you can add the particular reagent and run the particular chemical reaction. Yes, you will analyze the proteins, carbohydrates or the lipids and nucleic acid. Estimation or assay can be done. So generally the lipids are acid insoluble pool and they are the having higher molecular weight, and the lipids from the what you can say plasma membrane of the cells or from the membranes of the organelle. This is what we talk about a basic step of analysis of biomolecules. Well, in the next class, yes, we are going to discuss about how actually atomic absorption spectroscopy used to do the molecular analysis of the compounds, as well as we will discuss about primary and secondary metabolites also. Yes. Till then, take care, study well. Thank you very much.